Hello, my name is Saka Fajrian Febda Kurnia, and I am a master student at Sakura University for Biochemistry Department. For this experiment, I was helped by two supervisors from Biochemistry Department and also Food Engineering Department. This experiment called Some Phytochemical Properties of Spiraea japonica fortunei. For the graphical abstract, we could get the total flavonoids and phenolic and also the pH value from the extraction. Here, the outline will be introduction, objectives, materials and methods, results, and conclusion. First of all, I want to tell you about the free radicals. So, the oxidation reactions that occur constantly in our body trigger the formation of highly active free radicals that can damage cell structure and function. Free radicals are molecules that have lost their outer electrons that can react rapidly with surrounding atoms or compounds. I want to tell you about the plant that I used in the experiment. Genus Spiraea is belonging to the Rosaceae family, which comprises around 100 species. Species of Spiraea grow in moderate climate regions, especially in Asia. Spiraea japonica is a perennial shrub with pink flowers widespread across East Asia. It is the complex of 14 species that are morphologically distinguished depending on the leaf size and shape. The plant is known to contain active compounds such as alkaloids, flavonoids, and phenols. It has been used as a diuretic, detoxifier, anti-inflammation, pain reliever, and for the treatment of cough, headaches, and toothaches in traditional Chinese medicine for a long time. For the antioxidant characteristics, the general definition of an antioxidant is a compound that resists oxidation or inhibits reactions caused by oxygen or peroxides. Enzymes that are included as antioxidants are superoxide dismutase, glutathione peroxidase, catalase, thioredoxin reductase, and peroxyredoxin. Non-enzymatic antioxidant compounds work in four ways: free radical scavengers, transition metal chelators, oxidative enzyme inhibitors, antioxidant cofactors. Antioxidants and biological free radical scavengers can be classified as according to the targets, according to working mechanism, according to physicochemical properties. For the phenolic compounds, the antioxidant activity of phenolic compounds is achieved by reducing radicals to prevent adverse side reactions. Phenolic compounds can bind free radicals by transferring a single electron hydrogen atom from a phenolic compound to a single electron free radical. One of the most important properties of phenolic compounds is that they possess an antioxidant property that helps to prevent injury of tissues by removing the free radicals. Flavonoids. Flavonoids are the largest group of phenolic compounds found in nature. Flavonoids are polar compounds, so it is generally highly soluble in polar solvent. Flavonoids are found in all parts of plants, including leaves, flowers, fruits, and seeds. The presence of sugar attached causes them to be more soluble in water. Flavonoid compounds are antioxidants. They have been used as a component for drugs. Flavonoid can damage the bacterial cell walls and inhibit bacterial motility. Present investigation is undertaken by utilizing the plant Spiraea japonica fortunei with following objectives. First, the extraction of the flowers and leaves with different solvents. The second, preliminary phytochemical analysis. Third, determination of total phenolic and flavonoid contents at the different extracts. Fourth, evaluation the antioxidant activities. Collection of material and extraction procedure, Spiraea japonica fortunei was collected from the back garden of the Faculty of Engineering, Sakura University. Flowers and leaves of the plant were dried separately in shadow at room temperature. Then, dried flowers and leaves were grounded and then sieved to have a fine powder. Both methanol and ethanol extract of leaves and flowers were prepared. For this, powder samples were weighed and alcohol was added. The samples were stirred moderately on a magnetic agitator at room temperature for 24 hours for the extraction. Then, the solution was centrifuged and the supernatant was collected while the pellet was discarded. The clear filtrate was dried under a vacuum using a rotary evaporator. For the analysis in this experiment, we used Folin-Shio-Celsius method for finding the total phenolic compound, and also used aluminum chloride colorimetric assay to find the total flavonoids content. And also, we used the DPPH free radical scavenging activity to find the antioxidant value. Total phenolic contents of the Spiraea japonica fortunei extracts were determined by using the Folin-Shio-Celsius method. For this, the extract was mixed with an appropriate solvent, thiosulfate reagent, and distilled water. 3 minutes later, sodium carbonate solution was added and then allowed to stand for 1 hour in dark. At the end of the reaction, absorbance of the sample was measured at 765 nanometer by using a UV phase spectrophotometer. Aluminum chloride colorimetric assay was used for the determination of total sulfonate content. The extract was mixed with distilled water and sodium nitrate and left to stand for 5 minutes at room temperature. Then, aluminum chloride was transferred into the reaction mixture. After 5 minutes, sodium hydroxide was added. Finally, the volume was completed to 1 milliliter with distilled water, and for text, the intensity of the orange-yellow color created by the flavonoids was measured at 510 nanometer by using a UV phase spectrophotometer. Antioxidant activity was determined with TPPH radical scavenging activity with some modification. Various amounts of extracts were added to the test tube and the volumes were completed to 200 microliter with methanol. The samples were reacted with DPPH solution for 30 minutes at room temperature. Then, the decrease in the absorbance compared to control was monitored at 517 nanometer. Extraction yield is a measure of the effectiveness of the solvent and extraction method to extract certain components from the plants. Extraction yields of Spiraea japonica fortunei leaves were considerably higher than the flowers with both solvents. Total phenolic contents of the samples range from 11.08 to 25.64 mg GAE per gram dry matter, and the maximum value was obtained with methanol extracts of the leaves. The extraction with ethanol resulted in lower phenolic contents in the flowers and the leaves, which were in correlation with the extraction yields. The total flavonoid contents of the samples exhibited similar trends to the total phenolic contents. The lowest flavonoid content was observed at the ethanol extraction of Spiraea japonica fortunei flowers, while the highest was with methanol extract of the leaves. The results indicated that methanol is a better option for the extraction of both Spiraea japonica fortunei flowers and the leaves. Both total phenolic and flavonoid contents of the leaves were higher than the flowers, which could be attributed to the extraction yield, which was higher with methanol. The DPPH results also indicated that the use of methanol was more efficient than ethanol for extraction as with total phenolic and flavonoid contents. The conclusion that we could get from the experiment are, first, phytochemical screening showed the presence of various classes of bioactive chemical constituents in all extracts of Spiraea japonica fortunei, including phenolic and flavonoids. Second, total phenolic and flavonoid contents results show a large dominance in methanol extract. Third, the leaves of the plant are richer in bioactive compounds compared to the flowers. The last, both leaves and flowers have antioxidant activity. And that's all the data that I could share with you. Thank you for your attention and have a nice day.